Thank you very much, Costanza. Well, I am not that good researcher than Olga and you. I'm better to talking history, you know. And my talk will be, at least at the beginning, more a history about friendship, a journey that has been made especially from the Spanish organization that is also with me here today. We are really happy to stay here in Mitochondria, and you will know why. The history of the Spanish registry of primary mitochondrial disease, the first step was in 2016. Time has changed, and many of these people maybe you cannot recognize because 10 years after, also, you know, mitochondria is involved in aging. They have changed a little bit, as everyone's. And the history is that in Spain, they decided to start the registry because they saw that in Italy there was this initiative already ongoing, and they asked for an help from you. Then AEMI, that is the Spanish Association of Primary Mitochondrial Disease, with the Ana Carolina Diezmao, that is a foundation that helps them, reeling on the Mitocon experience and registry, Italian registry, involved two groups, that is the one led by Rafa Artuch, that presented yesterday, and one of the biggest and most historical group of Giulio Montoya, that is a long-time friends of Mitocon. But, you know, here something is missing. This some more Spanish-Italian people, that is me, from Madrid, that I'm now leading the clinical registry in Spain. And there we started 10 years ago. We started with a red cap registry that was just the same of the old Italian registry before that was moved to Genomit. But before entering patient and data, we had to build the network. And we started with a seed project trying to count the number of cases in the center in Spain, just collecting a few variables, and disseminate this initiative to the neurologists, pediatric neurologists, neuro-ophthalmologists, and experts in genetics and inherited metabolic condition. And this is a group that now is called MitoSpain, trying to work together as a multidisciplinary team. We had an incredible response. We cover all the 50 provinces in Spain, 50 centers, 9 lab and 41 clinical center answered to this first call we made. We also have made something similar to this Mitocon Congress. We have, it's my first time attending here, even if I'm worked also at Bambino Gesù Hospital in the past, but we have so in the last year with great attention to this Congress and replicate it in Spain the first time last September with more than 200 participants from more than 10 different countries, from Europe, from North America and South America. And now we are going to the result of the last, our work in the past 10 years. First of all, we have published the result of this call in 2020. We were able to recruit more than 2,000 cases. More of them, a large part, were patient carrying variants of the deafness associated with the susceptibility with aminoglucosides. When we remove these cases that are special, it's more a pharmacological susceptibility than a disease, we have 1,600 cases, mostly from mtDNA and 500 from nuclear DNA. What we understand at this point is that, as you can see, in 2014, 2014, for the nuclear genome, we have an increase of 5-fold number of diagnoses because of the implementation in Spain of the next generation sequencing. Why the mtDNA number of diagnoses are more stable, since Professor Montoya diagnosed the first case in the 90s, but as you can see, the last 20 years, the number of diagnoses is stable, really. And this is what's happened. We at the beginning, before the 2014, we were diagnosing the patient because of their phenotype. It can be a radiological phenotype, a Lee syndrome, or increased lactic acidosis with a high ratio, normal ratio lactate pyruvate to identify PDHA1 deficiency, for example, etc. And when we see the diagnosis made in the period before 2014, we see that we have just these 20 genes. And after, many new diseases appear. Sometimes disease in a type of patient that before we will not suspect a mitochondrial disease. For example, pure neuropathy, or patient with not this biochemical or clinical marker that are included in the famous Morava criteria, for example. We were also able to receive more cases also in the last 3 years, and they covered 10 years of NGS in Spain, showing and calculating the cumulative incidence in this period that actually in Spain there are 1 case for each 3,000 newborn. And as you can see, also the lower and the upper confidence interval is quite is small, because the quality of the data is high. There are more or less 120 new cases for each year in Spain diagnosed with primary mitochondrial condition. Of these, as you can see in this graph, we have 50% of the case due to nuclear DNA, the other 50% for mitochondrial DNA. For mitochondrial DNA, as you can see, the adults are more prevalent, as already know, and for nuclear DNA, it's more frequent, are more frequent in adults. There are some distinctive features. Everybody, when you are in the field of also the new drugs and new approval, sometimes company come to you and say, you know, you are expected to have 20 cases because of the incidence in Europe of this disease. And I say, and I usually use this registry to demonstrate that, well, in Spain we have some special feature. This TK2 higher incidence is a national feature, a distinctive feature. Is the same is for some variants as this mitochondrial ND1, the 3734AG that was demonstrated and described just in a Spanish family, causing loan, described by the group of Professor Montoya, that after this session will present also a specific Venezuelan variants, demonstrating that there are things that even in Europe can differentiate one population from another. We also understand that there are many N01 cases in each regions, and this is why, as Olga said, we need to put together case for some genes, try to understand the natural history, also even for new therapy, as is the case also from the Lee syndrome associated with ATP6 that we discussed yesterday. And now we are, in the last 2 years, we have left behind this epidemiological registry and starting a collaboration with the new whole genome sequencing project in Spain, a big project. I believe that you also have something similar in Italy. We have a national project that received many millions from this European funding after the COVID, in which we are performing 2,000 genome, and we have started a collaboration. Also because we just have found 150 genes out of the more than 300 genes that cause mitochondrial disease in Spanish population, and we would like to know why we don't found any of these genes, if we are properly diagnosing after the implementation of the NGS our patient, or we can do it better with the whole genome sequencing. These 2,000 unsolved cases were studied. Of these, before entering this genome project, only 5% of them were submitted to complete mtDNA sequencing. Here is the debate. Every patient with every phenotype deserves to have the complete mtDNA sequence. Well, let's see. Now it's NGS not even have a good capture of the mtDNA, while there is an advantage of the genome to have both the exome, the nuclear and the mitochondrial genome done. And 30% of the cases with this genome project has been solved in Spain. 12% that was for the exome reanalysis. And one of the problems we see that still there are places in which in the last 3-4 years have been submitted to virtual panels, because even if we have all the new advance in genetics, then the reality of the public system is that the resources are limited, and the genetic teams, they can do really complete exome in all the patient. But also some other cases were closed and solved because new amine gene were described, or the variants were reclassified. Other 18% of the cases were diagnosed with the whole genome sequencing because these new RNA genes, also intronic variant or complex CNV. But the most interesting thing for this audience is that 10 new cases with primary mitochondrial disease were also found there. And 3 cases were 3 patients with this new gene described a few years ago, associated also with one of the first steps of the primary CoQ10 biosynthetic pathway, and also 2 patients with mtDNA not previously covered by the study that the patient have been submitted to. And well, for example, in this ATP6 patient with a neonatal severe cardiomyopathy, he does not present with lactic acidosis. And then nobody asked for mtDNA in him. And well, maybe the genome will change the landscape, but not so much. Just 10 cases in all this year of unsolved case, to me, it's not a big change. We also identified the many carriers because all these people that were not submitted to mtDNA, and with the new result, we have so many carriers, and also it's useful for their family for genetic counseling. And now we are going to our future that is within the Genomit consortium. We entered the Genomit consortium, and now we had the foundings we have the foundings to create, we are on it, this biobank, in which in the last year we have included 100 patients with 100 sample, plasma, fibro, and cells, also to making omics, the metabolomics, proteomics, but also let's see if we can do also methylation for understand something. For example, with this biobank we have for for project. The one is to be everything's available to all the groups that want to collaborate and have fundings to make things. I believe that this project was, as like in Italy, was pushed and created by the patient organization, and we don't want to use this sample just for our group. We have these spirits to collaborate with everyone that have the capability to find a new treatment, because this is what we are aiming to. We are also assessing the new technology and using this for functional study, because we have to remember that many of our cases are not are diagnosed because of the clinics, but many of them have VUS, and we are trying to validate with functional study these variants. And then one of the main goals and idea we have within the Alla Mito Consortium is to validate biomarkers and find new biomarkers. And this also make this with omics. And regarding this, the use of omics and the biobank, I will present the two projects that we are going to do. One is with patients, because we have in Spain a very large cohort of PDHA1, more than 40 patients. In other countries, not so common. And as you know, this gene is a multi-enzymatic is a multi-enzymatic protein in which we have different subunits. One is sensitive to thiamine, but not all the variables are thiamine sensitive. The problem is that if you want to assess the thiamine sensitive, it's very expensive, and in few countries, few centers can do it. And then in the guidelines that are upcoming, I can anticipate that it will be suggested to use high-dose supplementation to all of them. But we already know that not all will respond. And I had the idea to see if using our biobank, our informed consent, our sample, we can use the sample collected longitudinally of plasma of this patient and see if a biomarker, when we use high dose, will suffer any change, both biomarker or proteomics or the metabolomics of these patients. Because in the registry, we are trying to make a longitudinal follow-up, both with samples than with the clinics phenotypes. The same is for this group of complex 3 genes, as is LIRM7, that you know that cause cavitated leukoencephalopathy, in which we had an experience that I will present the case of a child that had a spectacular response to ketogenic diet. But, you know, we are trying to demonstrate the relationship of this response with the diet to try to understand also the mechanism there. We have also diagnosed the far more family of far more affected individuals in this family that were previously treated and diagnosed as multiple sclerosis. And now we are trying to make also there a longitudinal follow-up with sample and also use the fibro to make some functional assay to understand the effect of ketogenic diet. The case that I follow in the last year is a case of all these points are lactic acid that, as you can see, started to growing as the patient started a clinical deterioration, suffering stroke-likes, vision loss, several stroke, coma, even DCA, the chloroacetate, was started to try to lowering this lactate, but nothing was helpful. She was admitted more than 200 days in a year, and the day after the ketogenic diet, the value dropped, and the patient is 3 years at home without any decompensation. And we are trying also some lesion in the UVI disappeared, the partially recovered the vision. This is also something that is common with the BCS1L, and both are gene involved in the complex 3, and we are trying with this follow-up and these samples and this fibro to understand if there is any pathway as this one, CMYC, that is sensitive to the ketogenic diet. Then the conclusion, the collaboration with MitoCon, we use you as an example of association. It has been very important for MitoSpain to grow, consolidate position at international level. Also, the partnership with Genomic was really important for us. Thank you very much, because it's really an opportunity to access fundings, idea and new opportunities. And now we are making our own projects as a group, also in this field of the omic and the registry with you together. Thank you very much.